All right, so welcome to the pre-lab for Lab 11, the isolation and identification of spinach pigments. In your lab manual, you actually have pictures of the spinach pigments. So there are several different ones. There's chlorophyll A, B, and these other ones, including beta-carotene, which you may have heard before. These structures are very common to how you would see structures drawn in organic chemistry. You'll notice that there appear to be some elements missing. So the way these structures are drawn, they are skeletal structures, and so every point is a carbon, and we know that all carbons have to have 4 bonds. So this carbon at the end here would then have to have 3 hydrogens bonded to it to give it the 4 bonds. This carbon here, which has 1, 2, 3 bonds to it already, would have 1 carbon to it. So if you take organic chemistry, you'll see structures like this a lot. All of the carbons and hydrogens have been shown on the pigment structures on page 1115 and 1116 of your lab manual. All right, so we are going to use a technique called chromatography in order to separate those pigments. So chromatography, some terms you should know with respect to chromatography are stationary phase, mobile phase, and RF value, right? So chromatography involves some kind of stationary phase. In our case, we're going to be using paper. And we're going to spot compounds on our paper, and then we're going to have some kind of solvent here. That's our mobile phase. We're going to put our paper into a chromatography chamber, and as the solvent gets pulled or moves up the paper via capillary action, what we'll see is our sample will get separated into the different pigments. So we're going to talk a little bit about chromatography and what causes the separation and how we can use this. So what causes the substance to move or stick to the paper? So paper is made of cellulose, right? And here's the structure of cellulose. Cellulose is a polymer, so it's a long chain, keeps going on and on like this. Remember these kinds of structures, so every point is a carbon. So here we have a carbon that has 3 bonds, so there's also a hydrogen to it. But you'll see lots of oxygens and hydrogens in this molecule, lots of OH groups here. And if we think back to our last lab where we looked at intermolecular forces and the one before that where we looked at polarity, right? We remember that these OH groups are particularly polar, right? This oxygen, very electronegative, pulls electrons away from the hydrogen and then the carbon here that it's bonded to, right? So we get these really strong areas of electron density. So if we looked at an electron density map, these oxygens would be very red, these hydrogens would be very blue. We have this very polar molecule. In contrast, here are our solvent molecules. So we have mostly hexane. Our solvent is going to be 90% hexane and a little bit of acetone, right? It's 10% acetone. So our solvent is mostly nonpolar. Carbon and hydrogen molecule is very nonpolar. Acetone is a little bit polar, right? We see this carbon-oxygen bond that's a little polar. So if we think back to our last lab about things that mixed well, right? If we looked at things mixing with water versus things mixing with oil, what types of things mixed well with water? Were they polar things, things like water, or were they non-polar things, right? Think similarly about oil. Okay, so we see our paper or our stationary phase is very polar, and our solvent is mostly nonpolar, maybe just a little bit polar. And so it's really the polarity, largely, also the size a little bit, but our pigments are all kind of similar in size. So it's mainly the polarity that causes this separation. So what exactly does this separation look like? Well, I'm going to show you a little video that's an example of separating inks using paper chromatography. So we're not going to watch this whole thing because we don't need to. But what happens is they're spotting different colored inks onto a paper. Right, let me get here. And then you can see they put the paper with those spotted inks into the chamber. The solvent is below that line, and it's going to start moving up the paper. And you can see as it moves up the paper, the colors start to move up the paper too. Right? And we're just going to speed this up a little bit because we don't need to watch the whole thing. But you can see that as they get further and further up the paper, you can see that the different colors that make up those inks have separated, right? So the different pigments that make up the colors and the different color markers have separated. And you can see that they travel at different distances, right? So this yellow one travels much further than this blue one, and this kind of pinky pigment down here just sticks right on the bottom, right? You can see how far the solvent moved. There's this line here. We call that the solvent front. So that's kind of what doing chromatography looks like as things separate. Right, so now we have our things separated, so what? We need to be able to compare them, right? And if we may have the same solvent and then same stationary phase, so same paper, but we may all have different sizes of paper, right? We may have longer ones or shorter ones. We may let the solvent front here move further or a shorter distance, right? So we have to be able to compare these things because we not only want to separate it, but we can use chromatography to identify the different types of things that are in our substance. So we do that with something called an RF value, right? And so the RF value, what we do is we measure the distance that the spot travels, right? And we measure it to the center. And we divide that by the distance that the solvent travels. So we're getting a ratio, right? And so it should not matter how long your paper is or how far your solvent is, right? If you have this substance A, you should get a similar RF value on different chromatograms as long as you use the same solvent and the same stationary phase, the same kind of paper. So you can use different kinds of stationary phases, you can use different kinds of solvents, and that's how we change our chromatography. Okay, so we figured out how to calculate an RF value. We've figured out how chromatography works to separate things based on polarity. It's really helpful when the substances we're separating are colored because then we can easily see them. It's possible to do chromatography with things that are not colored and use different ways to visualize the spots. But fortunately for spinach, our pigments are colored. Right? And they're colored because of something called conjugation. So you'll notice that when you look at the pigment structures on page 1115 and 1116 in your lab, you'll see lots and lots of double bonds, carbon-carbon double bonds, right? And when we have this pattern of a carbon-carbon double bond and then a single bond and a double bond and a single bond, we say that these things are conjugated, right? As compared to something that doesn't have these alternating single and double bonds. Right, and so this molecule here is actually colorless. It has no color, whereas this molecule here, when we have all these alternating single and double bonds, right, it has a color. And this conjugation, if you've talked about sigma and pi bonds, right, occurs because in a conjugated system, you have every single carbon, whoops, let's go back, sorry, every single carbon here is sp2 hybridized, right? So every single carbon has a p orbital on it. And so electrons can travel all the way down this chain as long as we have this conjugation, right? As soon as we have one of these sp3 carbons in the middle here, like this, we break that conjugation, right? But this whole segment of this molecule here is conjugated, right? So electrons can travel that whole distance. And this conjugation gives rise to compounds being colored for some reasons that we're not going to go into right now. All right, so that takes us to the lab. So what are we going to do today? We're going to determine whether frozen spinach has the same nutritional value as fresh spinach. That's our overall goal. What we're going to do is we're going to separate the pigments from spinach using paper chromatography. There should be a Y on that. So we're going to separate it using paper chromatography. We are providing you with some pictures of that and a little video. You're going to calculate RF values from those pictures, and then you're going to use electron density surface pictures, which we will provide for you, and compare the polarity of the pigments, and then use, that should just be one, and use the IMFs to identify each pigment in the chromatogram. All right, so if you haven't already done your pre-lab, you should definitely do that. For part A, you're going to watch the lab video posted on Blackboard showing how the spinach pigments were separated using chromatography, and then you're going to complete table 11.1 for the frozen and fresh spinach using the pictures posted on Blackboard. For part B, you're going to determine the polarity of the pigments. You're going to use electron density pictures and the images posted on Blackboard, as well as the spinach chromatograph pictures, and the information about the structures of the paper and the solvent to answer the questions in this section. The last one being you're actually going to compare the frozen spinach and the fresh spinach and decide whether you think the nutritional value is the same and why. All right, so as a reminder, if you have any questions, be sure to contact your lab instructor if you need help with something, and good luck.